Hello, class 10th. In the previous video of how do organisms reproduce, you studied about why do organisms reproduce. And you were also introduced with the term asexual reproduction and its types. Now, in this video, we'll study the different types of asexual reproduction in little bit detail. The first is fission. Now, unicellular organisms undergo binary fission, that is, to divide a single cell, that is the mother cell, into two daughter cells. Now this splitting of cell into two daughter cells can take place in any plane. There are different planes of cell division in unicellular organism. Like in amoeba, the splitting of two cells during cell division, it can take place in any plane because it does not have a fixed shape. But for the organism which has a fixed shape, for example, if we talk about leishmania, which cause kala azar, which has a whip-like structure, the division takes place at one end of the cell. So this has a definite orientation in relation to its structure. In some unicellular organisms, for example the malarial parasite Plasmodium, the nucleus of the cell divides and redivides to form many daughter cells. And these daughter cells are surrounded by a protective covering called as cyst, which later on burst when the favorable conditions arise. This is called as multiple fission. Fragmentation. Now, in some multicellular organisms which have simple body organization, for example, Spirogyra, they simply breaks into smaller pieces on maturation, and these pieces or fragments grow into new individuals. Regeneration. Now, some multicellular organisms which have well body differentiation, which has tissue or organ system level of organization, they regrow their body from the cut body part. For example, if somehow their body part is cut into small pieces, so those different pieces can regrow their lost body part to form a new individual, to form a separate individual. This takes place in Hydra and Planaria. This is known as regeneration. Regeneration is actually carried by specialized cells. These cells proliferate and make large numbers of cells. From this mass of cells, different cells undergo changes to become various cell types and tissues. These changes take place in the organized sequence referred to as development. Now the question is, how is fragmentation different from regeneration? If you look at Spirogyra, it has a very simple body design, although it is multicellular. So the different pieces break and they grow into new individuals. But it is not true for all multicellular organisms, because their body is made up of specialized cells which organize into tissues, and tissues organize into organs. So therefore multicellular organisms use more complex ways of reproduction. In case of planaria and hydra, there are specialized cells, as we have discussed earlier, which helps in regeneration. Budding. Organisms like Hydra use regenerative cells for reproduction in the process of budding. Now, in Hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. These buds develop into tiny individuals and, when fully mature, detach from the parent body and become new independent individuals. Vegetative propagation. There are many plants in which parts like the root, stem, and leaves develop into new plants under appropriate conditions. This property of vegetative propagation is used in methods such as layering or grafting to grow many plants like sugarcane, roses, or grapes for agricultural purposes. Plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds. Such methods are also make possible the propagation of plants such as banana, orange, rose, and jasmine that have lost the capacity to produce seeds. Another advantage of vegetative propagation is that all plants produced are genetically similar enough to the parent plant to have all its characteristics. Buds produced in the notches along the leaf margin of bryophyllum fall on the soil and develop into new plant. This is the vegetative propagation in case of leaves of bryophyllum. Spore formation. You must have observed the thread-like structures in the bread mold, rhizopus. They are not the reproductive parts. The tiny blob on a stick structures are involved in reproduction. The blobs are sporangia, which contains cells or spores. The spores are covered by thick walls that protect them until they come into contact with another moist surface and can begin to grow into a new rhizopus individual. Tissue culture. In tissue culture, new plants are grown by removing tissue or separating cells from the growing tip of a plant. The cells are then placed in an artificial medium where they divide rapidly to form a small group of cells or callus. The callus is transferred to another medium containing hormones for growth and differentiation. The plantlets are then placed in the soil so that they can grow into mature plants. Using tissue culture, many plants can be grown from one parent in disease free conditions. This technique is commonly used for ornamental plants. This was all in the asexual reproduction, and in the next video, we'll shall start with sexual reproduction.